Man versus microbes? Man and microbes. Centuries ago, when science took a backseat to superstition, infectious diseases must have been terrifying. They were a sign of supernatural powers or the wrath of God. Robert Koch unequivocally showed that a disease, anthrax, was caused by bacteria, and we were enlightened. We now know that Yersinia pestis was the probable cause of the Black Death. Variola and measles viruses contributed to the decline of the Aztec civilization. Mycobacterium tuberculosis is the cause of the disease known in the 19th century as consumption. Indeed, we have an astounding depth of understanding about the diversity of infectious pathogens. We are aware of the mechanisms they use to counter host defenses and cause disease. Yet this knowledge only partially mitigates the threat of infectious pathogens. Unfortunately, diseases once eradicated still remain a concern. The potential risk of the intentional release of infectious pathogens necessitates vigilance and preparedness. Man lives in a state of precarious equilibrium with the microorganisms that surround him. Most of the time, the relationship is surprisingly harmonious. Young and healthy humans come in contact with the bacteria or viruses native to their region. They develop a natural resistance to them by doing so. Illness occurs when this natural resistance no longer balances the virulence of the bacteria. Occasionally, the balance of power between microbes and men is upset on a much wider scale. A foreign germ may get imported to a region where it was unknown. A native virus may develop a mutant strain. The result may be an epidemic. They could be due to migrations, wars, social upheavals or undernourishment. Many people think of bacteria, viruses, and other microorganisms as our enemy. This perspective, called the germ theory of disease, was first proposed in the 19th century. It maintained that illness springs from the actions of infecting microorganisms. It draws battle lines between us, the afflicted hosts, and them, the invading microbes. Germ theory was crucial in identifying many diseases caused by microbes. It helped us to find ways to prevent them through immunization, sanitation, and improved living conditions. By the mid-1960s, many experts concluded that infectious disease was all but conquered. Microbial world. Researchers were told to shift their focus to chronic medical conditions such as heart disease and cancer. Today, many scientists recognize the need for a more ecological view of the microbial world around us. Microbes and their hosts, including humans, ultimately depend on each other for survival. But this optimism was shaken in the mid-1970s and early 1980s by the onset of the following: Lyme disease, Legionnaire's disease, toxic shock syndrome, and human immunodeficiency virus/acquired immunodeficiency syndrome, HIV/AIDS. They dealt further blows with the development of antibiotic-resistant bacteria, the appearance and spread of severe acute respiratory syndrome, SARS, H1N1, Middle East respiratory syndrome, coronavirus, MERS-CoV, Ebola, and finally COVID-19. Scientists began to re-examine the relationship between hosts and microbes. To shape our future health, we should learn from the microbes that accompanied us on our evolutionary journey. It is human nature to want to know where we came from. Individually, we investigate our family lineages to discover ancestors lost to history. Collectively, scientists examine data from a vast array of sources. They range from ancient fossils to current genomes. It was to determine where humanity itself originated, and how we came to be who and where we are as a species today. For that reason, in recent years, researchers have paid much more attention to our second genome, that of our microbiota. Our microbiota is all of the microscopic organisms that live on and in us. They play a role in our digestion, train our immune system to correctly respond to pathogens. They manufacture key vitamins and taking up space that could otherwise be exploited by pathogens. Gut microbes are the worlds within worlds that have evolved alongside us, their hosts. They are our early human ancestors, who moved from place to place, ate new foods. They had encountered new animals and environments. Our current microbiome, the collective genetic material of our microbiota, reflects some of that deep history. We can glean information about human history from those organisms within us in several ways. One is by using the parts of our own cells that are, in essence, microbial, our mitochondria. These organelles can be considered extreme symbionts. They are remnants of microorganisms that once lived free. But they are now integral parts of all eukaryotic, complex, cells, producing energy and regulating metabolism. Mitochondria. Mitochondria retain their own DNA, separate from that of the cell's nucleus. For many types of research, this mitochondrial DNA is preferable to nuclear DNA as an object of study. Analysis of in the 1980s led to the conclusion that humanity originated in Africa. It dates back to a common maternal ancestor somewhere around 100,000 to 200,000 years ago. Today, many scientists recognize the need for a more ecological view of the microbial world around us. Microbes and their hosts, including humans, ultimately depend on each other for survival. And although the microorganisms that cause disease often receive more attention, most microorganisms do not cause illness. In fact, many of them protect us, helping our bodies function properly. They compete with harmful organisms in an ongoing contest for habitable space in and on our bodies. There is a close connection between microbes and humans. Experts believe that about half of all human DNA originated from viruses. They infected and embedded their nucleic acid in our ancestors' egg and sperm cells. Microbes occupy all of our body surfaces, including the skin, gut, and mucous membranes. In fact, our bodies contain at least 10 times more bacterial cells than human ones. It blurs the line between where microbes end and humans begin. Microbes in the human gastrointestinal tract alone comprise at least 10 trillion organisms. They represent more than 1,000 species. They are thought to prevent the gut from being colonized by disease-causing organisms. Among their other beneficial roles, microbes synthesize vitamins. They break down food into absorbable nutrients, and stimulate our immune system. The vast majority of microbes establish themselves as persistent colonists. They thrive in complex communities within and on our bodies. In many cases, the microbes derive benefits without harming us. In other cases, both host and microbe benefit. And though some microbes make us sick and even kill us, in the long run they have a shared interest in our survival. For these tiny inhabitants, a dead host is a dead end. The success of microorganisms is due to their remarkable adaptability. Through natural selection, organisms that are genetically better suited to their surroundings have more offspring. They transmit their desirable traits to future generations. New generation. This process operates far more rapidly in the microbial world than in people. Humans produce a new generation every 20 years or so, bacteria do it every 20 to 30 minutes, and viruses even faster. Because they reproduce so quickly, microorganisms can assemble in big numbers and variety in their communities. If their environment suddenly changes, the community's genetic variations make it more likely that some will survive. This gives microbes a huge advantage over humans when it comes to adapting for survival. We assume that just about any bug we are saddled with can be wiped out with a quick round of antibiotics. But in the US alone, roughly 2 million people every year get infections that can't be treated with antibiotics. In that, 23,000 of them die. The bacteria to blame, so-called superbugs, are now present in every corner of the planet. About 50% of people infected with K. pneumoniae or E. coli bacteria won't respond to our most powerful antibiotics. That means doctors are increasingly running out of the ammunition they rely on to fight these harmful microbes. Bacteria have been evolving to resist the drugs designed to kill them since the first antibiotic was discovered in 1928. But our overuse of antibiotics in farming, prescription drugs and antibacterial soaps has supercharged the process. Before antibiotics, something as minor as a cut or a sore throat could be a death sentence. The measures to contain antibiotic misuse are small steps in the right direction, but much more needs to be done. No amount of technological wizardry is likely to succeed in wiping out the microbes that can cause disease. They are too prolific, too adaptable and too widespread to disappear from the face of the earth. Nor can vaccines supply the whole answer. So many kinds of bacteria and viruses exist and it is impossible to vaccinate people against all of them. We could succeed against smallpox as only one strain of the virus is known. The pneumococci and streptococcus bacteria have at least 100 different strains. A vaccine against just only one of the strains would be next to useless. Some germs develop new strains that are unaffected by existing vaccines and drugs. The greatest pandemics of our times, the influenza to COVID-19, have produced mutant strains. The mortality they have caused is also appallingly high. The microbes have powers that remain beyond the magic of medicine. Thanks to drugs, man's burden of illness is greatly reduced. But microbial diseases are a constant threat and will remain so as long as microbes exist.